Welcome to the educational video lecture series by MedDevice Circle. This video lecture series is made in collaboration with Anamol Laboratories Private Limited. In this video lecture series, we are discussing the fundamentals of clinical chemistry test, which are very frequently used in IVD industry as well as in pathology laboratory. So without wasting any time, let us go back to our slides. Friends, please allow me to share my screen. I hope my screen is visible to everyone. So as I discussed, friends, in this video lecture series, we are only discussing the fundamentals of clinical chemistry test, which are very frequently used in IVD industry as well as in pathology labs. This entire video lecture is divided into 4 different modules. In the first module, we covered the basics of photometry, where we discussed what is wavelength, what are complementary colors, how Beer-Lambert's law got formed, and how we implement in our day-to-day life to generate patient reports. What are the components of colorimeter and what is the difference between colorimeter and spectrophotometer? In the second module, which we are discussing today, we are discussing what is the role of enzymes in diagnostic test and in general, what is the role of enzyme in our IVD industry. Here we discussed what are the role of enzymes, what are the factors that affect the enzyme activity. And we also discussed what are the different types of biochemical reactions that happen in pathology laboratory. All these videos are in our previous video lecture series. In this particular video, we are discussing how to read any IFU by any company for any product. Friends, here IFU means instructions for use. So without wasting any time, before we start our today's lecture, let us quickly revise whatever we have discussed in our previous video lecture series. In our previous videos, we discussed that enzymes are nothing but they are catalyst. They are proteins which play the role of catalyst in our body. They do not take part in the reaction, but they enhance the speed of the reaction. The way enzymes function is by binding to a particular substrate, forming an enzyme substrate complex, and then converting the substrate into product and releasing the product back into the system. This step is critical to us because this is how our majority of the biochemical reactions happen in human body. We also saw that because enzymes are catalyst and they do not take part in the reaction, therefore the quantity of enzymes are the same before the reaction and after the completion of the reaction. Therefore, it is not clinically relevant for us to identify the concentration or the quantity of enzymes. But what is important is we have to identify the speed of enzyme or the activity of enzyme. Because most of the diseases happen because the activity of enzyme either increases or decreases. The activity of enzyme has certain key attributes. Number one attribute is enzyme is very specific to a particular substrate. This is why we use enzymes in our biochemical formulations also. Secondly, enzyme has catalytic efficiency, which means that enzyme lowers the activation energy which is required to start the reaction. And this is how enzyme actually starts the reaction. Third, enzyme activity can be measured. So either we can measure the amount of substrate consumed per minute or we can measure the amount of product formed per minute. And lastly, enzyme activity depends on several environmental factors. Now, what are the environmental factors that affect the enzyme activity? We discussed that in our day-to-day pathology lab, there are 5 major environmental factors that affect the enzyme activity. The first one is the pH of the solution. Second, the ionic strength of the buffer. Third, the reagent to sample ratio, because when the reagent to sample ratio changes, it actually affects the enzyme kinetics. Fourth, timing of the absorbance, whether are we taking the absorbance in lag phase of the reaction or we are noting the absorbance in the log phase of the reaction. And fifth, temperature. if we increase the temperature of the reaction or decrease the temperature, how it affects the enzyme kinetics. All of this we discussed in our previous video. We also discussed in our previous video that there are basically three major types of biochemical reactions. First is the endpoint reaction, where we wait for the reaction to get completed and then take the absorbance. Here, we take only one absorbance to measure the enzyme activity or to measure the substrate concent or the analyte concentration. Secondly, there is a second type of biochemical reaction, which is kinetic reaction. In kinetic reaction, we don't wait for the reaction to get complete. We take the measurement of the absorbance while the reaction is still in progress. So here, we take minimum two absorbance. This is very, very important because this is relevant for all the enzyme activity measurements. If we have to do enzyme activity estimation, we follow kinetic procedure. But if we have to calculate the concentration of analyte, then we follow the first method, which is endpoint reaction. Fixed time kinetics is also a type of kinetic reaction only. The only difference is that in fixed time kinetics, the 2 points where we take the absorbance measurement are fixed. We cannot change these 2 points. Whereas in normal kinetic reaction, we can change these 2 points of measurement as long as both the points are in log phase of the reaction. Now, in this particular video, we are going to understand that how we can read the pack insert or the instructions for use by any company for any product, as long as they are in the clinical chemistry segment. So let's dive deep into this particular lecture. So friends, we all know that every company, when they supply a product, they will give a pack insert along with the product. Now, pack insert is nothing but it's like a small paper which comes inside the kit. This paper or this pack insert will give us information about how the product is to be used, how the solution needs to be performed or prepared, how the reagents are supposed to be stored, how the calculation needs to be done, what kind of precautions do we need to follow, what is the principle of the product, what are the range of normal values as per this particular test. All this information are given in the form of instructions for use. Some people call this IFU as pack insert. Some people call this IFU as literature. Some people call it as kit literature. But essentially, all these terminologies are the same. They are meant for the user or the consumer. Whoever is consuming the test, whoever is using the kit, that particular person needs to read and understand this pack insert before using the product. Now, each company has a different way or different style of putting the information. We need to understand how we should read this information. Now, just for an example, we have taken one pack insert of a product called creatinine from Anamol Laboratories. Now, as you can see in this window, there are multiple information that is given in the pack insert. Now, which information is relevant to us, which we should not skip before performing the test. We will focus on that particular information, because if we don't have knowledge about these informations, then either we will perform the test incorrectly, or we will not be able to perform the test. So let's try to dive deep into what are the information that we need to follow or we need to understand. The first information that we need to understand is the kit components. Friends, whenever we buy a diagnostic kit, when we open the box, there are certain components which are given inside the box. We need to understand what are these components, how many different types of components are there. We need to understand what is written on the label of the component, and we need to understand why this particular component is given inside the kit. If we do not understand this, we will not understand how to perform the test. So in this particular example, when any user purchases a creatinine kit from Anamol, then the kit contains three different bottles. First bottle is the picrate reagent, second bottle is the diluent reagent, and third bottle is the creatinine standard. The value of standard is 2 mg per deciliter. Now, friends, why this is important to us? Because if we purchase the kit and we see that as per the literature, there has to be three different components inside the kit. But when I open the kit and I only find two components, the third component is missing. Will I be able to perform the test? The answer is maybe yes, maybe no. If I am able to perform the test, will I be able to report the test or will I be able to generate the patient report? Maybe yes, maybe no. So we need to compare that whatever the manufacturer is claiming in the pack insert, whether those components are practically and physically present in the kit or not. Second thing that we need to understand is how to do reagent preparation. Now, friends, whenever we open the diagnostic kit, it is not necessary that the reagents which are given in the diagnostic kit, they are ready to use. That means we just open the bottle and straight away take the reagent in the test tube for reaction. It is not necessary. Sometimes what happens is the reagents which are given in the kit are not ready to use. They have to go through a process which is called as reagent preparation. Now, reagent preparation is nothing but preparation of working solution. Because we have discussed in our previous lectures that when we take the reagent in the test tube and when we add patient sample into the test tube, the sample and the reagent will start the reaction. Now, what is this reagent? This reagent is not the reagent which is given in the kit. This reagent is called as working solution. Now, working solution may be readily given in the kit as a ready to use form, or it may not be ready to use. It may be in separate components. We have to mix these components and then only working solution will be ready. So this information needs to be understood before performing the test. This information is given in the reagent preparation section. Now here you can see that when we saw the kit components, we saw that there are two different reagents in the kit. One is reagent 1, which is picrate reagent, and one is reagent 2, which is the diluent reagent. Now, when we understand reagent preparation, we see that we have to mix the picrate and the diluent reagent in 1:1 ratio. Now, what is this 1:1 ratio? 1:1 means if we take one part of picrate, then we have to take one part of the diluent and we have to mix it. So say for example, we have 10 patient samples that we need to perform today for creatinine test. Now we know that for every patient sample, we need 1 ml of working reagent. So if we have 10 patient samples, so we will need 10 ml of working reagent. Now, if we have to prepare 10 ml of working reagent, then we need to take 5 ml of picrate and mix it with 5 ml of diluent. This is 1:1 ratio, equal parts. Then only our working solution will be ready when we mix both these reagents. Now, this particular section also talks about how do we store the working solution and what is the stability of the working solution. Here it is very clearly mentioned that the working reagent is stable for 6 months at 2 to 8 degrees Celsius, which means that when we mix the picrate and the diluent and when we keep this working solution at 2 to 8 degree, it is stable for up to 6 months. And here also it is mentioned that the kit should be stored at room temperature and the kit is stable for expiry date, which means the shelf life of the kit is, for example, 2 years. Then the kit is stable for 2 years, provided it is stored at room temperature. But when we mix the picrate and diluent in 1:1 ratio, and then we prepare the working solution. This working solution cannot be kept at room temperature. The working solution has to be kept at 2 to 8 degree, and it is stable for up to 6 months only. It is not stable up to 2 years, unlike the kit. So that is why we have to clearly understand how do we prepare the working solution. For some products, we have to prepare reagent 1 and reagent 2. We have to mix in 4:1 ratio, which means that we have to take 4 parts of reagent 1 and mix with 1 part of reagent 2. Like for example, SGOT or SGPT. In these particular products, we have to mix reagent 1 and reagent 2 in 4:1 ratio. So for one patient sample, if we use 1 ml of working solution, then we have to take 800 microliter of reagent 1, and in that we have to add 200 microliter of reagent 2. Then we will get 1 ml of working solution. So how to prepare working solution? The instructions is given in this particular section: reagent preparation, storage, and stability. Now, next to this, there is a third information that we need to understand. This information is called as test parameters. Some companies write as assay parameters. Now, test parameters and assay parameters are the same. In this section, you will understand how to program the software of the autoanalyzer before using the test. Here you can see that multiple information is given, like what name you have to add in the program, what is the reaction type, what is the reaction direction, what is wavelength, and so on and so forth. Now, when we program any semi-auto analyzer, or for that matter, whenever we program a fully auto analyzer, we have to add certain information in the software. This information is given in this particular section of the IFU. So test parameters are nothing but the set of information that needs to be added in the software to program the analyzer. Next information that we need to follow is the procedure. Once we prepare the working solution, how do we really do the test? This information is given in something called as assay procedure. Now, you can see in this particular video that in assay procedure, there are two different test tubes which we have to take. One is the test tube of standard and one is the test tube of test. For standard, we have to add 1000 microliter of reagent, which is a working solution. And in that, we need to add 50 microliter of standard. In the test, we need to take 1000 microliter of reagent, which is the working solution, and we need to add 50 microliter of patient sample. Then we need to mix it in the above mentioned ratio and start the stopwatch. And then we need to aspirate the mixture into the flow cell and measure the absorbance at 30 seconds and 90 seconds. So this exactly is the procedure of performing the test. Now, it is important to note here that in some parameters, when we do the color development, like for example, a parameter of glucose, whenever we do the color development of glucose, the final color is not stable for a very long period of time. The color may be stable for up to 8 minutes or 30 minutes or 60 minutes or 120 minutes. So that particular information of final color stability is also mentioned in assay procedure. So if the manufacturer claims that the final color stable is up to 120 minutes only, then we need to take the absorbance within 120 minutes after the incubation is complete. All of this information is available in the instructions for use. The next section that we need to understand is the reference value. This is the normal range or the abnormal range of the patient sample. Now, every manufacturer will tell you that the laboratory has to decide its own normal range and abnormal range. But sometimes when the laboratory does not have a normal range or abnormal range, then The manufacturer also tells the normal range and abnormal range in the form of reference value. These reference value can be added into the software while doing the programming of the parameter. So friends, if you understand these 5 information which is given in the instructions for use, you can very well use this test. You can very well use the kit and you can very well generate the patient report. But these are, if you fail to understand any of these 5 information, then either you will not be able to do the programming or you will not be able to perform the test. Or if you are able to perform the test, you will not come to know whether the test you have performed is correct or incorrect. That is why it is important to understand minimum this 5 information in the pack insert. Now, different manufacturers have different way of designing the pack insert. Some manufacturers put X information on the top and Y information in the bottom, whereas some manufacturers may put X information on left and Y information on the right. We just have to locate where this information is printed in the pack insert and read that particular section so that we understand that this information is written in so and so area and this is what I need to follow. Friends, I hope this video has been helpful for you. If you have any kind of questions or if you have any kind of suggestion for us, please write in the comment section and this will help us to improve our future videos. Thank you very much, friends, and I will see you in the next video.